Hello, bachcho. Let's see the question. So the question says Taq polymerase, which is used in amplification of DNA, is related with. So इन चारों ऑप्शन में से हमें वो आइडेंटिफाई करना है जहां टैग पॉलीमरेज को हम यूज करते हैं. So the key concept in this question is to understand what is taq polymerase. So first of all, taq polymerase is a type of a DNA polymerase. So DNA polymerase is an enzyme which catalyzes the synthesis of a DNA strand by nucleotide addition. Now, this Taq polymerase, DNA polymerase enzyme is a very special type of enzyme because it is thermostable enzyme. So, what is a thermostable enzyme? A thermostable enzyme is that enzyme that can withstand very high temperatures. Now, this Taq polymerase is derived from an bacteria which is known as the Thermus aquaticus. So, Thermus aquaticus is a bacteria which is found in very high temperatures, and therefore this DNA polymerase helps in the synthesis of new nucleic acid strands in the Thermus aquaticus bacteria. So this Taq polymerase enzyme is derived from this bacteria. Now this Taq polymerase is utilized in a process which is known as polymerase chain reaction, also known as PCR in short. Now, this enzyme plays a very important role in polymerase chain reaction, and with the help of polymerase chain reaction, DNA amplification takes place. Now, if we see the basic steps of PCR, then multiple copies of the target DNA sequence can be made with the help of PCR. So, in the first step, it involves denaturation of the target DNA sample. Now, this step takes place at a very high temperature of 94 to 96 degree centigrade. So, this high temperature is required for separating the two strands of the DNA sample, that is for the denaturation of the DNA sample. The next step is annealing of the primers. So a primer is designed which is complementary to the target DNA sequence, and this primer anneals to the separated DNA strand at a temperature of 52 to 60 degree centigrade. Again, this is much higher temperature from the average body temperature or the room temperature, that is 52 to 60 degree centigrade is the temperature at which the primers will anneal with the target DNA sequence. Now this high temperatures is required because we need to keep the two strands of DNA separated, that is denatured, because if the temperature comes to room temperature, then that will cause in the re-annealing of the target DNA sample. So we don't want the target DNA sample to re-anneal. Instead, we want the primer to anneal to the target DNA sequence. Therefore, we need a higher temperature. The third step in this process is the extension. So the primer is extended by with the help of the enzyme, that is Taq polymerase. So Taq polymerase will extend the primer by addition of the nucleotides complementary to the target DNA sequence, and this takes place at a temperature of 72 degrees centigrade. So now we can see that all steps in the PCR, so all steps in PCR takes place at a much higher temperature than the average or the optimum temperature which can be withstanded by a normal DNA polymerase enzyme. Therefore, we utilize the enzyme Taq polymerase for this process because Taq polymerase can withstand much higher temperature, and this temperature of PCR which is required for the process to take place can be withstanded by the Taq polymerase enzyme. So the Taq polymerase can withstand this high temperature, and therefore this enzyme is particularly used in the process of DNA amplification, which is polymerase chain reaction or PCR. So if we come back to the question, the first option says hybridoma technique. So hybridoma technique is not used in DNA amplification, and neither is Taq polymerase used in this enzyme. So this won't be the answer. PCR technique, as we just talked about, that PCR is the DNA amplification technique where we use Taq polymerase as the enzyme, the thermostable enzyme. And the third option says gene cloning. So in gene cloning, we do not need Taq polymerase because in gene cloning, we do not require a very high temperature. In fact, in gene cloning, we require enzymes like restriction endonucleases and DNA ligases. Taq polymerase is not required. And in the fourth option, we have recombinant DNA technology. Again, in recombinant DNA technology, we do not require Taq polymerase, but instead we require a restriction endonucleases, which plays an important role in recombinant DNA technology or the rDNA technology. So our final answer for this question is option 2, that is PCR technique. So Taq polymerase is the enzyme used in PCR technique. Hope you understood this well. Best of luck. Thank you.